Hello, everyone. My name is Corinthia Brown, and today I will be talking about my research that I conducted over this summer, analyzing ligand specificity to assess the evolution of TLR4 and creating a drop-in cassette. I performed my research in the Harms lab under the guidance of Kona, Orlandi, and Mike Harms. So that title was probably a lot to take in, so let me start to break things down. First, TLR4, toll-like receptor 4, is a protein, a molecular structure encoded by DNA that performs various bodily functions. Often when proteins don't work or overactivate, we end up having health issues. TLR4 is a protein that activates a portion of our immune system, inflammation. Inflammation is a vital process that our bodies use to remove foreign entities and help restore function to damaged tissue. However, when inflammation excessively activates, it can lead to arthritis, neurodegeneration, and sepsis, which contribute to 11 million deaths a year. Inflammation results from inflammatory cytokines produced by the NF-kappa B pathway, activated by the toll-like receptor 4. We know that liposaccharides, LPS, present on gram-negative bacteria, drive the dimerization of TLR4 and activate inflammatory cytokine production. However, we do not know the rules that govern TLR4 activation, making it impossible to control the production of those inflammatory cytokines. Now, to understand why TLR4 activates, the Harm lab studies the alterations and functions throughout the protein's evolutionary history. We look at the effects of altered DNA on ligand specificity, which determines what binds to TLR4, activating the inflammatory pathway. In addition, we also look at the level of activation that is achieved to determine the physiological significance of the resulting signal. By comparing changes in the proteins encoding DNA to what is produced or how much signal is produced, we can start figuring out those rules. Understanding these rules allows us to transfer data collected from model species such as zebrafish to applications in humans. In addition, the data from these experiments helped us to understand what changes in amino acid sequence have on protein function. However, when the TLR4 in question is far enough from humans on the phylogenetic tree, we experience difficulties observing the protein's activation. To observe these distant species TLR4 activities, we developed a method that allows the outer portion, the exodomain of a protein, to bind to its specific ligand, yet have an inner portion, a terdomain, that allows for interactions with human proteins that bridge a connection between the TLR4 and NF-kappa B pathway. So our hypothesis was that intercellular protein interfaces have diverged between species, which means frogs TLR4 expressed in human cells cannot activate inflammation because it cannot interact with human immune signaling proteins. So if you look at this diagram down here, you can see the green down here is the frog TLR4, and it has this kind of circular immune signaling protein. Whereas humans are represented by this blue protein that has square immune signaling proteins. And when they interact with these downstream pathways, such as the NF-kappa B, they end up resulting in inflammation, which is what we are studying. On the other hand, if we place them into human cells and they have these signaling proteins that are square and cannot interact with the TLR4 in question, we end up not having any activity at all. So to test this hypothesis, I'm making a cross-species hybrid of TLR4 proteins where the extracellular domains of different species can be fused to the intercellular domains of human TLR4. This should maintain the intercellular human protein interactions that are required for human cells to initiate inflammation. So as you can see, the frog, the green protein, now has this blue inner domain that has the squares and can interact with the human cells' immune signaling process. So as you can see, it can interact with the square ones and create inflammation. Now, to obtain the inflammatory response from TLR4, we must place it, transfect it, and its co-receptors ND2 and CD14 into HEX cells, human embryonic kidney cells, the human cells that we mentioned in the previous slides, in order to express the downstream NF-kappa B pathway. Now, this pathway has been engineered in these cells to express runilla and luciferase, which can then be measured with a spectrometer. Once we understand that and to what extent TLR4 is activated, then we can correlate where the species and most recent common ancestors lie and the variations in structure and activation to make a hypothesis for ancestral reconstruction, where we can then test these areas of interest by making point mutations and studying the resulting activity in the hex cells. So basically, my goal in the lab was to be able to make a method of measuring these species that had previously shown too small of a signal. And to do so, we tested our theory by placing the human endodomain portion onto the frog TLR4 exodomain, but we didn't see much of an increase in signal. Then, thinking that perhaps we had altered the manner in which the co-receptor and TLR4 could interact, we tried shifting the exodomain helix by 4 to 5 amino acids to see if the manner in which the protruding alpha helix coming from the transmembrane domain was situated made a difference in the resulting activity of the protein. As you can see, it didn't make enough of a difference to actually be able to observe the frog TLR4 signal without having to worry about the significance. As we saw some difference between the plus K and del TI mutants, we decided to see if helix shifting was a concern for other constructions that would be made in a similar way. And so basically, when I say helix shifting, I'm saying that you see this orange portion here, that's like the terdomain, and it's represented by these couple of amino acids. And alpha helix generally compose the transmembrane domain because it is this hydrophobic portion that is kept away from water, and the most favorable conformation of amino acids for that area is going to be an alpha helix. So basically, we can say, oh, by shifting it 1, 2, or 3 amino acids, it might be having this completely different effect out in this red area where MD2, its co-receptor, binds, which would have an incredible effect on the specificity and activity levels of the protein. So, and with that in mind, we tested this theory by repeating these mutations in highly responsive human TLR4, adding and taking away amino acids from around the transmembrane domain. We found that shifting the helix didn't really have an effect on the activity levels, with no mutant showing the higher signal levels we were looking for. It was interesting to note that the hydrophobic regions of the protein, like within the transmembrane domain, when they were altered, showed a large decrease in signal and an increase in signal when only the buffer was present. This would imply that if we were to have impacted the hydrophobic portion of the transmembrane domain, we would generally see constitutive activity from the damaged cell. And then another aspect of my project was to explore the homologs of TLR4, which had evolutionarily split off from the currently operating TLR4 we had already noted within our lab. So zebrafish TLR4-BB and zebrafish TLR4-AL are both duplicates of the zebrafish ancestral TLR4, which have now become inoperational. It would be nice to be able to see which ligand version of LPS that they responded to help create an idea of what the ancestral protein may have looked like. But even with placing the human TER domain on them, I got very little signal and not enough to be significant in telling us what it responds to. On the other hand, we also have CD14, which was tested and showed very little response despite having a human TER domain placed on it, as well as having its respective co-receptor, MD1. MD1 is a homolog of MD2, which was the co-receptor used in all other experiments, as MD1 typically doesn't work with any of the other constructs. So going forward, I have a couple things that I've been working on in lab, getting the mutagenesis to work. I have the Sicilians, which are like kind of an amphibian, but not quite. And so they might be able to tell us about that ancestor between humans and amphibians, even though frog has not shown any signal. In addition, pike may be able to tell us about the other duplicates of the zebrafish TLR4 that we couldn't get a significant enough signal from. And then from there, once we get that data, we will be able to test the ancestors and see what these proteins have basically turned from, and that'll be able to be like applied in medical uses and transferring the data from these model species into humans. So that ancestral thing is a little ways off still. So what is directly next for me going into the fall is testing mutants that may alter TLR4 activation in zebrafish. So basically, my fellow lab mate Sophia has some ideas on what mutations in TLR4 and its co-receptor MD2 have outsized effects on specificity and signal. These hypotheses have come from both data collected in our lab and notes about the protein's respective structure in these two papers listed below. And so far, I have one of the constructs done and I'm working on the others, so we'll be able to see that pretty soon. Thank you for listening to me talk about my summer research, and have a wonderful day.